Hello everyone and welcome to week 2. This week we will focus on chemical neurotransmission. This is the basis of our understanding of psychopharmacology and our focus for the remainder of this class after this week. We can also see that neurotransmission is the communication tool of the brain and is the link between the anatomical structure that houses the billions of neurons and the behaviors we see manifest both in our clients and in ourselves. So for the overview of what we're going to be discussing, we'll be looking at the link between last week, where we discussed brain structure and development and chemical neurotransmission through the nervous system function. We'll mostly be focused on the chemical neurotransmission and then launch into our study of psychopharmacology. The reading this week was from both the handbook, and this is how I'm going to refer to the required text by Preston. It's the one that is the Handbook of Clinical Psychopharmacology for Therapists. So that will be referred to as Handbook. And then we're also reading Chapter 1 from Stahl, which is an excellent resource. It's on reserve at the library, and it's kind of the gold standard of psychopharmacology. It's used more probably when one wants to understand things to the degree of prescribing. But I really find the images and the information in that book helpful as well. So in the handbook, there's a small section on brain structure that provides a review and a link from week 1 to week 2. There's also a great figure that correlates brain structure to function and to then psychopathology. And there's a little overlap in what is covered on neurotransmission, but the Stahl chapter will be our focus. And our main focus, as I said already, is chemical neurotransmission, and this is going to lay the foundation for our study of psychopharmacology, which is introduced in Chapter 4 of the handbook. The big picture that we're looking at here is the nervous system, and this gives context to our study of the central nervous system and the neurons and the chemical soup per se that is occurring there with the different neurotransmitters. I bring this overview picture in because I want us to remember that there are other neurons. For example, there are sensory neurons, and you can see that down there in the somatic sensory receptors, where touch, pressure, heat, these things are sensed by the peripheral nervous system and send a message to the brain in the central nervous system and then elicit a response. There's also motor neurons, which you can see on the other side down in the bottom there, where there's smooth muscle, cardiac muscle, etc., that also elicit a response by sending messages to the central nervous system. And this is important to refer back to when we consider side effects of some of the medications. For example, the extrapyramidal side effects such as tremor or tardive dyskinesia, which is described as irregular jerky movements, excuse me, are associated with first-generation antipsychotic medications such as chlorpromazine that works by blocking dopamine. And so therefore, as we move forward, just remember that the effects of the medication is not just within the brain. It can affect different parts of the nervous system, and thus that's where we get some of the side effects that we see with medications. Another representation of this overview is here in this slide. Again, it's breaking down the nervous system into the peripheral and the central nervous system, and of course we're focusing on the brain. And as we focus on the neurons and the communication system of the brain, we'll see that sometimes the psychopathology is related to a malfunction of a specific brain structure. Example of this is in OCD, there's thought to be issues within the cingulate of the limbic system. However, we can't take this as a hard and fast rule. There are sometimes more than one structure involved. For example, schizophrenia is thought to be related to issues within the prefrontal cortex, the septum of the limbic system, and or the reticular system of the brainstem. So there can be more than one structure, and then also sometimes the organic nature of the mental imbalance is hard to correlate with a specific anatomical structure. So structure, function, and the psychopathology aren't always related. It's not always a clean-cut correlation. But this is interesting, and there is a chart in the handbook on this that you can look at that shows the relation between those. Here you can see that we're breaking down the anatomy of anxiety, and this was a great image that I found online, and I've put the reference there at the bottom. And this shows how the structure and function of the brain can correlate with anxiety. So, for example, and you can't see it, that it didn't transfer in the image, so that little black box that the eye is seeing, in the image that I found, there's actually a picture of a gun. So, of course, yes, very scary stimulus there. So the body, the eyes, sometimes there might be smells. So the stimuli is taken in, and then the thalamus breaks down the message contained in the stimuli, and then the cortex can translate and process that message. Then the amygdala gives it an emotional significance, and then if we look specifically at, I think it's number 6 there, the bed nucleus of the striata terminalis perpetuates this fear response, and the locus coeruleus holds the autonomic response, which then creates the fast heartbeat, the increased blood pressure, and the entire experience is then held in the memory of the hippocampus. So there are correlations between the anatomy and what is occurring within our emotional response. So we can see then how structure, function, emotional response are all correlated, and the communication of all of this is occurring within the brain structures via neurons. So that gets us down to this neurocommunication or neurotransmission and the signaling that occurs both within the brain and within the communication between the peripheral and central nervous systems. When looking at the neurons, we can think of this as a forest within the brain. There are billions of neurons in our brain, and in this lecture today we're going to take a closer look at neurons and the foundation that this lies for our study of psychopharmacology. So as you all know, I love the example of a tree, so I was very excited when I found this journal cover from Cell with a tree on it representing a neuron, and in a sense the soma, or the body, could kind of be pictured there as like the center of the tree, and the dendrites and the dendritic spines that thus reach out receive information from other neurons. So if you think of this as like the leaves and the branches of the tree, it can feel the sunlight, the wind, maybe animals that have taken residence in its branches, and so it's sensing all of this information and then feeding that into the soma, or the body, the central core of the neuron, and then that then perpetuates a signal to the next neuron through the axon, and in that picture it would be the tree trunk. And we could think of this as the communication within the roots, or within the earth. And there's we can consider then how that perpetuates the response and continues the communication on. I also enjoyed this picture here of the little guy walking through a neuronal forest. So there's a bunch of neurons, and this man's trying to navigate his way through. And then the top middle picture there is just shows how the complexity of all the dendrites and then dendritic spines can become quite complex in how much they've connected and communicated with other parts of the neuronal tissue. So we just break down and look at the neuron itself. We're back to the image I left you with last week that shows the anatomical structure here. As we've stated, the dendrites are taking in information and the axons are communicating to the next neuron. So if you look at figure 1.1 and 1.2 in the stall chapter, you can see an expansion on this image. And the communication between neurons can take place at the dendrites, the cell body, or the soma, which is the center part there, and the axons. So communication can occur at all three of those locations. And as we look at neurotransmission, we're considering both the anatomical and the chemical aspects. So both the structure and the signals that are occurring within this neuronal communication. And Stahl uses the example of a road map, and I really like that. If we think about the neurons themselves and their connections to each other as being a very complicated roadmap, then we can consider the signals, or the neurotransmitters, as the cars, the people, the buses, bikes that travel along and that communicate thus from one neuron to the next. So that example may or not work for you. We also want to recognize that the neuron produces both proteins and neurotransmitter to elicit these signals. So to look at that in a more specific slide, I found this online, which really clearly showed the excitation-secretion coupling. So the red part is the presynaptic neuron, and the tan part there is the postsynaptic neuron. And we're seeing the communication from the one to the next. And the action potential, which is the electronic impulse that comes down the axon into the axon terminal, which is the bulb part there with all the vesicles in it. And what happens is that the calcium channel there's a shift in calcium influx, and that thus releases the neurotransmitters. And the neurotransmitters in this picture are the little green circles, and you can see that as number 3 there. Those little neurotransmitters are then released into what's known as the synaptic cleft, and that's that space between the presynaptic and postsynaptic neuron. And then the neurotransmitter will bind to receptors in the postsynaptic neuron, and then that then elicits a signal within the postsynaptic neuron to do whatever function that specific signal has. And so there is this excitation of the presynaptic neuron, a secretion of neurotransmitters, and that together creates this communication from one neuron to the next. So what are these neurotransmitters? Those little green dots there can be represented by numerous different chemical structures, and this is the chemical soup that we often hear referred to, because all these different structures are floating around and creating different signals. Things like adrenaline, which I'm sure you're familiar with that, or dopamine and serotonin, endorphins, glutamate, acetylcholine, the purple one there is GABA aminobutyric acid, also known as GABA, and these are all the different chemicals that are creating these signals within the neurotransmission. And there's a little bit more information in figure 4C, D, and E in the handbook, and we'll also look at these in more detail as we study each molecule in the course. So neurotransmission. This is where our communication tool, and it's how the neurotransmitters elicit their response from one neuron to the next. And this communication method alters what is happening at the postsynaptic target neuron in classic neurotransmission. However, there's also retrograde and volume neurotransmission. And in retrograde, the postsynaptic neuron actually has communication back to the presynaptic neuron, so it's not just a unilateral flow. And then in volume neurotransmission, there's a diffusion of the neurotransmitters that occurs onto other neurons, and this can contribute to what we think of as this soup. So it's not always a direct hit. It's not like it's just always going from point A to point B. There can be some peripheral communication happening that's not always what's intended. And so you can see some more about these in these. I've noted the figures from Stahl that these different neurotransmission types correlate to. And it's important to realize that every step in these processes is a potential point at which medication can modify neurotransmission. So there are, we'll talk about things like reuptake inhibitors and things of that such nature. So we'll get into more detail as we talk about the specific drugs. When we look just at classic neurotransmission, this is where we can see that the electrical impulse, which is just held within the one neuron itself, comes down the axon, creates that excitation secretion coupling that we saw a couple of slides ago. So that's this here. And then the neurotransmitter is released into that synaptic cleft that we discussed. And it can be any one of those neurotransmitters or a combination thereof. And then there's an influence on the postsynaptic neuron. And so this is what we consider classic neurotransmission. Now what happens once those neurotransmitters hit the postsynaptic neuron is often referred to as signal transduction. And this is a cascade of events that follows that stimulation of the synaptic receptor. And we can explore these more as we look at different medications and whether they're G protein linked neurotransmitters, ion channel linked neurotransmitters, whether there's hormonal communication or neurotrophin creating these different messages. So these are just examples here, and these are the four main types of signal transduction. And we can think of something called the Pony Express, and Stahl refers to this, and it's a relay of messages. So there's, or you can even think of it as a relay race, where one individual passes the baton to the next individual, and that perpetuates the communication. And we can also think of our roadmap analogy and having a set of directions. And at any point in those directions, something could go wrong. So even though we have an intended route along our roadmap, we could take a wrong turn, there could be an accident, there could be a detour, a signal could be out, and all these potential issues along the route are points where mental illness or imbalances can occur, and thus, since these are points of potential malfunction, it's also a point where psychotropic drugs may be of help, and we can kind of think of them as the road crew kind of coming to try to fix things. Ultimately, the impact is on the genes, creating a direct link to mental illness and correlates to the function of the psychotropic drugs. And so when we look at the genes and we talk about this, we're looking at the relation to what is happening within our client's neurons. The presynaptic genome is communicating with the postsynaptic genome, which boils down to the ultimate communication that's happening within the nervous system. So this shows how malleable our nervous system can be and the effects of environmental influences. So stress, abuse, diet, our nutrition, daily activities, therapy, drugs, both drugs of abuse and helpful drugs, medications, can then change the gene expression that's happening within neurotransmission, and genes can be activated and silenced, and therefore we're really influencing the epigenetics, and this is where our treatment of mental illnesses and psychiatric disorders come into play, and we're seeing the connection between the biology and the psychology of our clients. So where does pharmacology come into play? While we dive into the nuts and bolts of medications, we can also keep the perspective that the client stressors, whether they're good or bad, influence outcomes as well. So I have a cartoon here that says this antidepressant works best if you take it with water lapping near your hammock on a Caribbean beach. So environment's important, and then also, as we talked about, the genes and the specific biology is important. So there's a lot coming into play when we consider the impact of treatment on our clients. So how do these medications affect the body, and how does the body affect these medications? And so that's what we're looking at with pharmacodynamics and pharmacokinetics. When we think of the dynamics of the drug, it's how it influences the body, and a bulk of our discussion as we go through each module will talk about how these medications work. And then with the pharmacokinetics, we're looking at how the body functions interact with the drug. So we're looking at absorption, distribution, metabolism, or for example, there's also enzyme systems within the body that can inhibit and or induce the breakdown of medications. So for example, certain drugs, if there's other drugs that are present, that can influence the absorption, distribution, or metabolism of the drug. And then there's also some foods, such as grapefruit juice, that can have specific impact. So we'll discuss that as those things arise with each medication. Here I have a little example of how psychopharmacology has been around for a while. This is a little ad for Coca-Cola when it still contained properties of the cocoa plant. Before all these fancy drugs we have today, there used to be quote-unquote brain tonics like this one that use a little of nature to help with nervous afflictions. And so I just thought this was a little interesting caveat there that, this is early development of psychopharmacology. And students often wonder why there can't be a perfect drug. And Preston explains in the handbook this really well at the end of chapter 4, that basically there's no ideal drug because medications typically bind more than one receptor. So you have medications having effects at receptors beyond what the intention is. And this also relates to those charts in chapter 4. So, for example, there are more receptor subtypes of serotonin than one would necessarily have known initially. So the response that one wants might be for just one specific subtype of the receptor, but the medication then impacts all receptor subtypes or multiple receptor subtypes. And the receptors also exist in more than just one desired target of the brain. So I bring all of this up because it's the messy nature of these medications that relate to the unwanted side effects that often make medications used within psychopharmacology difficult to tolerate, and thus leads to medication non-adherence, which we will also discuss. So there's not really an ideal drug, but the researchers are continuing to work on drug development. And so I just wanted to point out that there is a long road to reach the ultimate product of a drug, and there's still medications coming onto the market that are improved. For example, the atypical antipsychotics that we use today are better tolerated and used more often than the first generation antipsychotics, which I discussed earlier have the extrapyramidal side effects. So there's still lots of research going on and new medications evolving that are often quite interesting and often do have improvements with their side effect profiles. So where do we head next? We want to understand the influence of medications on the neuroanatomy of the brain and how they elicit their action, and their impact on our clients as we wade through the numerous medications on the market so that you have some familiarity with what is being used in treatment. And we will try to put the pieces of the puzzle together in our client's treatment and road to well-being. So I hope that was helpful. I know that was definitely an overview, and we will get into some more details in our discussion and as we launch into our modules on the specific disease states and the medications used to treat those. All right, thanks so much, and I look forward to our discussion.